Factors affecting vaccine protection. The level of protection afforded by vaccination is affected by many genetics and environmental factors, including age, maternal antibody levels, barrier antigen exposure, vaccine schedule, and the vaccine dose. Although most of these factors can't be readily modified, age of vaccination and the schedule of vaccination are important and key factor in planning immunization program. The vaccine dose is established during early clinical development based on optimal safety and immunogenicity. However, for some populations such as older adults, a higher dose might be beneficial, as has been shown for influenza vaccine. Moreover, intradermal vaccine has been shown to be immunogenic at much lower fractional dose than intramuscular vaccine for influenza, rabies, and HB vaccine. Age of vaccination. The highest burden of and mortality from infectious diseases occur in the first 5 years of life, with the youngest infant being most affected. For this reason, immunization programs have largely focused on the age group where there is the greatest benefit from vaccine-induced protection. Although this makes sense from an epidemiological perspective, it's somewhat inconvenient from an immunological perspective, as the induction of strong immune response in the first year of life is challenging. Indeed, vaccination of older children and adults would induce stronger immune response, but would be of little value if those who would have benefited from vaccination have already succumbed to the disease. It's not fully understood why immune response to vaccine are not as robust in early infancy as they are in older children. One factor which is increasingly well documented is interference from maternal antibodies acquired in utero through the placenta, which might reduce antigen availability, reduce viral replication in the case of live viral vaccines such as measles, or perhaps regulate B-cell response. However, there is also evidence that there is a physiological age-dependent increase in antibody response in infancy. Furthermore, bone marrow niche is to support B-cell are limited in infancy, which might explain the very short life immune response that are documented in the first year of life. For example, after immunization with 2 doses of capsular group C meningococcal vaccine in infancy, only 41% of infants still had protective level of antibody by the time of booster dose administered 7 months later. The case of T-cell independent antigen, in other words, plain polysaccharide from HIV, typhoid causing bacteria, meningococci, and pneumococci, animal data indicate that antibody response depend on development of the marginal zone of spleen, which is required for the maturation of marginal zone P cells, and this doesn't occur until around 18 months of age in human infants. These plain polysaccharide vaccine don't induce memory B cell, and even in the adult provide protection for just 2 to 3 years, with protection resulting from antibody produced by plasma cell derived from marginal zone B cells. However, converting plain polysaccharide vaccine into 2 cell dependent protein polysaccharide conjugate vaccine, which are immunogenic from 2 months of age and induce immune memory, has transformed prevention of disease caused by the encapsulated bacteria, pneumococci, HB, and meningococci over the past 3 decades. These are the most important invasive bacterial pathogen of childhood, causing most cases of childhood meningitis and bacterial pneumonia, and the development of conjugate vaccine technology in 1983 has transformed global child health. Immune response are also poor in the older population, and most of the vaccine used in older adults offer limited protection or a limited duration of protection, particularly among those older than 75 years of age. The decline in immune function with age, known as immunosenescence, has been well documented, but despite the burden of the infection in the age group and the increasing size of population, has not received sufficient attention so far amongst immunologists and vaccinologists. Interestingly, some have raised the hypothesis that chronic infection with cytomegalovirus might have a role in immunosenescence through unfavorable effect on the immune system, including clonal expansion of CMV specific T cells population known as memory infection and reduced diversity of naive T cells. In high income countries, many older adults receive influenza, pneumococcal and varicella zoster vaccines. Although data showing substantial benefit of these vaccines in past few decades in the oldest adult more than 75 years of age are lacking, however, emerging data following the recent development and deployment of new generation high dose or adjuvanted influenza vaccine and an adjuvanted glycoprotein varicella zoster vaccine suggest that the provision of additional signal to the immune system by certain adjuvants such as ACE-S01, MF59 can overcome immunosenescence. It's now necessary to understand how and why and to use this knowledge to expand options for vaccine-induced protection at the extreme of life.